Dear students, in the last video, we were discussed about the central dogma of molecular biology. In this video, we have to discuss in detail the DNA replication. You know, each and every organism produces young ones of their own kind. Not only the higher organism, the plants, the microorganism, very small, minute, microscopic organism produces or that undergoing division that produces the new cells, new organisms. How it is possible? It is because of the ability of the DNA to replicate or to duplicate and produces its replica and transmit the character to progeny. What is DNA replication? How it replicates? So to discuss in detail the DNA replication. The ability of the DNA to produce the exact copies of itself is called DNA replication. It is also called DNA duplication. In which stage of cell cycle the synthesis of new DNA takes place? You all know about the cell cycle. In a continuously dividing cell, you can divide it into division phase. Actually, the young phase, mitosis or meiosis, where phase, anaphase, metaphase, anaphase and telophase. The cell divides into two. We can see four stages: the prophase, metaphase, anaphase and telophase. But in the interphase period, again we can divide it into G1, S and G2 phase. So synthesis of new DNA strand takes place in the S phase of cell cycle. Before the division of cell, prior to the division of cell, the synthesis of new DNA takes place, and the newly divided DNA segregates to sister chromatids and separated and goes to daughter cell. So in the S phase of cell cycle, the synthesis of DNA takes place. The parental DNA strand unwind that open and act as a template for the production of new strand. By using this template, the complementary new strand is synthesized. So that is the daughter strand. In the daughter strand, you can see the complementary nucleotide as present in the parental strand. In each and every replication, one is the parental strand, one is the daughter strand, namely synthesized strand. And this type of replication, which conserves the parental strand in each and every replication, that is called semi-conservative method of DNA replication. Method is also called semi-conservation method or semi-conservative method of DNA replication. In 1958, Matthew Meselson and Franklin Stahl did an experiment on E. coli, Escherichia coli, and confirmed the replication of DNA is semi-conservative. They proved the replication of DNA is semi-conservative. Meselson and Stahl did the experiment on E. coli bacteria, Escherichia coli. So this experiment is also called Meselson and Stahl experiment. Meselson and Stahl experiment. Franklin Stahl. So it is also called Meselson and Stahl experiment. So Meselson and Stahl grew E. coli bacteria in a heavy isotope of NH4Cl in the nuclear media of NH4Cl, N15, NH4Cl. It is not radioactive isotope. A heavy isotope of nitrogen, that is NH4Cl. In this NH4Cl media, when they grew the bacteria, E. coli bacteria, Escherichia coli, so all of its DNA, it incorporates the nitrogen in the double-stranded DNA of this E. coli bacteria, all such N15 NH4Cl molecule, that is nitrogen source, is incorporated in the DNA. In the DNA strand, you can see N15 as the nitrogen source. N15, both of these two strands incorporate this N15 for its nitrogen bases. All of its nitrogen base incorporates the N15 in its double-stranded DNA, because it is a source of nitrogen. N15, a heavy isotope, is the source of nitrogen. All of its nucleotide which contain the nitrogen bases incorporates the N15 nitrogen, because they grew in the N15 NH4Cl media in several of generation, and they differentiate the N15 DNA from the normal DNA by a centrifugation process. In the centrifuge tube, if you centrifuge the double-stranded DNA, in the cesium chloride, cesium chloride density gradient centrifugation gradient. So these heavy isotope containing DNA is extracted from the E. coli bacteria and centrifuged. They settle at a particular region. So in this particular region with heavy density, so it is called the heavy region. Heavy density, we call it as H region. That settle in the H region of centrifugation tube. When the DNA extracted from E. coli bacteria after growing in several of generation in N15 media, the centrifugation with cesium chloride density gradient, the DNA settled at heavy region because it contained N15 NH4Cl, heavy isotope. So settle at heavy region, the density gradient is heavy. Then they transferred the bacteria, E. coli bacteria, into a different media which contains N14 NH4Cl, a normal isotope, light region. So in this, after 20 minutes, the bacteria divides. E. coli bacteria divides in every 20 minutes. After 20 minutes, the DNA duplication occurs, DNA replication occurs. The two strands of DNA get separated. These two strands of DNA get separated, but the available nitrogen source in this media is only N14. Newly synthesized DNA strand incorporate N14 as a nitrogen base in the nucleotide. So one is the parental strand and one new strand was synthesized. New strand incorporates N14 for its nucleotide or nitrogen bases. When they centrifuge this extract from the E. coli bacteria, they got a hybrid region. The light and heavy isotope of double-stranded DNA set at a different region. It contains heavy isotope because it contains N15 nitrogen, and also, so this one is N15, and also it contains N14 in its strand. One of the strand contains N14 isotope and nitrogen bases, and another strand that contains N15. So you can see the N14 here. So N14, a light DNA settled at the median position, and heavy strand that also settled the same because it's a double stranded. It forms a hybrid density. The hybrid density contains both of these two strands, the heavy strand and light strand. Both of these two strands are present, so it forms a hybrid density in the density gradient cesium chloride solution. They allowed it to replicate the E. coli bacteria. Nearly about 40 minutes, it doubled once again. Every 20 minutes, E. coli bacteria doubles. After 14 units, it duplicates and produces a new strand. These two strands get separated. Act as a template for the production of new strand. These two strands get separated. The parental strand. These two are parental strand. Newly synthesized strand that contain only N14. N14 as a source of nitrogen. N14. So this N14 NH4 media only the nucleotide contain N14 normal isotope. So this N14 containing newly synthesized strand or new strand gets synthesized by using the parental strand as a template. So this one is the newly synthesized strand. All these newly synthesized strand contain N14. This one is the newly synthesized strand. Newly synthesized strand that contain N14. This one is the parental strand. Newly synthesized strand contain N14. Newly synthesized strand N14. So this one is the newly synthesized strand that contain N14. Which one is the parental strand? These two are parental strand that contain N50, a heavy isotope. But these two are the parental strand, even though it is a parental strand that contain N14 only. So one of these two strands contain N14 in its double-stranded DNA. It contains N14 and N50. This contains N14 and N50. Both of these two strands contain N14. Nitrogen is both of its strand. So when they were centrifuged this extract from E. coli bacteria, they observed the hybrid DNA that settled at one position, the hybrid DNA molecule. The two strands are present, the heavy strand and the light strand. The heavy strand and light strand both are present. It forms the hybrid region. And two newly synthesized strand that is light that contain N14 as a light isotope that settle at light region in the centrifugation tube that settle at light region. I can label it as L, light region, the H. This one is the hybrid region. So hybrid and light region and light density comes in equal proportion. It confirms that in each and every replication, one of the parental strand is concerned in the daughter. So the type of replication in DNA, each and every DNA replication, one of the parental strand is concerned in the daughter. One newly synthesized strand. The type of replication is called semi-conservative method of DNA replication. Semi-conservation method of DNA replication. Each and every replication, one of the parental strand is conserved in the daughter, one newly synthesized strand. So that is the semi-conservative method of DNA replication. How actually DNA replicates in the cell? So only after the replication of DNA, the chromosomes get separated and goes to daughter cell. Cell divides into two organisms are produced in unicellular organism. But in higher organism, the growth, development, the formation of new organ, new structures, all are formed by cell division only. For cell division, DNA must be replicated. How DNA replication takes place? So, what are the enzymes are involved in the process of DNA replication? We have to study in detail the DNA replication, or it's also called duplication of DNA. The replication or duplication of DNA is semi-conservative. In the S phase of cell cycle, the DNA replication takes place. So, prior to the DNA replication, we can see the specific protein that identifies the region of DNA replication. In the double-stranded DNA, a specific point or specific region where the DNA replication starts that is identified by a specific protein. An enzyme called helicase. Helicase is the that cuts the double-stranded DNA, that breaks the hydrogen bond. This enzyme is called helicase. Helicase is the enzyme that identifies a specific auric site or origin of replication, auric site in the double-stranded DNA, where the guidance enzyme comes and attaches, breaks the double-stranded DNA. The double-stranded DNA open up. The double-stranded DNA open up Y-shaped fork. So this Y-shaped fork is called replication fork. This 4 point. This is the 4 point. So it is called replication fork. Replication fork. So this one is called the 4 point. The parental strand that run in the 5-3 direction. 5-3 direction. Another strand is in 3-5 direction. 3-5 direction. The opposite, complementary and anti-parallel. So DNA dependent DNA polymerase. DNA dependent DNA polymerase is the enzyme. Polymerase is the enzyme that joins new nucleotide and synthesizes the new strand of DNA. But the source of energy like ATP molecule, ATP, CTP, GTP, TTP, the source of energy that is adenosine triphosphate, cytosine triphosphate, guanosine triphosphate, thymine triphosphate. So all these are source of energy which initiate or which activates the nucleotides. So after the activation of nucleotide, the DNA polymerase enzyme that initiates the synthesis of new strand complementary to the parental strand. The new strand is synthesized which is complementary to the parental strand. New nucleotide comes in this region and that is joined by phosphodiester bond, and new strand is synthesized in 5-3 direction. It synthesizes in 5-3 direction with the help of DNA dependent DNA polymerase enzyme. This DNA dependent DNA polymerase enzyme brings new nucleotide which is complementary to the template strand. The parental strand acts as a template copy. Xerox machine li hage original ಮೇಲ್ಗಡೆ ಇಟ್ಟು ಕೆಳಗಡೆ Xerox ತಗೋತೇವೆ, ಇದು original copy ಇದೆ, ಇದು Xerox copy, but it is a complement. It is slightly different from the Xerox and original. So we can get the complementary copy. If adenine is present, thymine is present, adenine comes, guanine is present, it's a new nucleotide is cytosine. Cytosine is present, new nucleotide is the guanine. So new complementary nucleotide comes, it forms the phosphodiester bond. DNA polymerase enzyme forms the phosphodiester bond and it leads towards the 4 point. So this is called the leading strand. So one run towards the leading strand, that is called the leading strand. So it is a continuous strand. It is also called a continuous strand, or it is also called leading strand. But in another strand, it has to wait for the opening. The primer is formed and new DNA synthesis or new strand of DNA synthesizes in 5-3 direction. So in the opposite strand, and again the primer is formed. Here also the formation of primer, you can see the formation of RNA primer before the synthesis of new DNA strand. So smaller fragments of DNA were formed that run in opposite direction, that is in 5-3 direction. So the smaller fragments of DNA were formed in the opposite strand that lag behind in the process of replication. So this strand is called discontinuous strand. This one is a continuous strand, this one is discontinuous strand. Or it's also called lagging strand. That lag behind in the process of replication. The smaller fragments so formed are called ophajanic fragments. These fragments fragments are called Okazaki fragments. So these Okazaki fragments are joined later by using an enzyme called DNA ligase. DNA ligase is an enzyme which acts as a glue or stitcher. So that's also called molecular stitchers, which joins the Okazaki fragment that forms a continuous strand of DNA. And nuclease is an enzyme that gives the proofreading. So topoisomerase is an enzyme that relieves the stress after unwinding process. We can see the complete synthesis of new strand. So that means in the double-stranded DNA, the parental strand open up and new strand is formed. That is, the daughter strands are formed. Daughter strand into the parental strand. So in each and every replication, one of the parental strand is conserved in the daughter. So the method of replication is called semi-conservative method of DNA replication. After complete replication, at specific point where specific nucleotide is present, the releasing factor, a type of protein that stops the synthesis of DNA, and it is twisted in a right-handed fashion that forms the double helical model of DNA. So that's about DNA replication. Thank you. Thanks for it all.